안녕하십니까. 제가 이게 사회학하는 직분을 맡은 지는 제가 상당히 오래됐는데, 저는 대구 경북대학교 병원의 정형외과 소속이고요. 이 재생의학을 우리 임정욱 교수님하고 같이 접하고 있는 사람 중에 하나입니다. 그래서 이번 기술 교류 회의를 이제 국내 오거나이저로 제가 맡고 있습니다. 그래서 먼저 바쁘신 중에도 오늘 참석해 주신 여러 해외 연자분들과 또 참석해 주신 많은 분들께 감사를 먼저 드립니다. 먼저 이게 제가 이거 한국말도 해야 되고 여기서 대사를 적어 주셨거든요. 한국말도 해야 되고 영어도 두 가지를 다 해야 됩니다. 그래서 먼저 이게 혹시라도 핸드폰, 휴대폰 진동 모드로 또는 무음으로 먼저 해주시면 이제 행사 진행에 도움이 될 것 같습니다. Good afternoon, everyone. I am Chang Go from Gyeongbok National University Hospital. I'm doing the orthopedic surgery. I'm an orthopedic surgeon, and I'm also organizing this 9th XLab Day event. And I'm your moderator from the domestic in Korea. And I also would like to introduce the international organizer, Professor James Yu. Yeah, would you mind to, yeah. Yeah, thank you very much, Professor Yu. He is from the Wake Forest University, and he has a role of international organizer. He helped to make this program successful today. And before we beginning, please ensure that your mobile phones are set to the vibrate or the silent mode to avoid any disruption during the event. Then now we would like to begin the BrainLink XLab Day Talk concert. We would like to try to watch this movie.
Amidst the world's rapidly accelerating changes and competition, what kind of future are you preparing for? We are charting a path to the future through science and technology exchanges, bolstered by international cooperation. In this era, where scientific and technological competitiveness is directly linked to a nation's diplomacy and security, the power to overcome such challenges lies in international cooperation that fosters synergy through collective efforts, and from talents who will shape the forthcoming era. The scholars of the world are gathering in Korea. A platform that facilitates exchanges between Korean and international scholars, as well as between seasoned and emerging researchers, is taking shape. BrainLink XLab Day, organized by the Korean Federation of Science and Technology Societies, aims to be a global hub of talent. By consistently enhancing and expanding the global talent network, we're laying the groundwork for a brighter future. By inviting global scholars, XLab Day provides an opportunity for young Korean researchers to glean insights from the vast knowledge and experience of both Korean and international scholars, as well as to engage in collaborative research. BrainLink XLab Day establishes a global science and technology network among overseas scholars and Korean researchers. In 2024, BrainLink XLab Day will be joined by globally renowned scholars, including Professor Luke Pyeong-se Lee of Harvard Medical School, Professor Andrew Goodwin of University of Oxford, Emeritus Professor Teruo Okano of Tokyo Women's Medical University, Jeffrey Newmark of NASA Goddard Space Flight Center Research Astrophysics, and Professor Dan I. Anderson of Uppsala University. The discussions will span 12 national strategic technology fields, pivotal to Korea's advancement, including the three game changers: quantum mechanics, nuclear technology, and AI. The event will see the participation of 40 Korean and international scientists and engineers. With 2 organizers leading each technical segment, the gathering will foster in-depth academic exchanges, encompassing the sharing of recent trends and information in core technology fields, in-depth discussions on research findings, and the sharing of ideas. And talk concerts will be held in collaboration with all BrainLink XLab Day sessions. The focus of XLab Day is connectivity and sustainability. Following the event, communities and clubs will be established based on each XLab Day session to ensure ongoing dialogue among attendees and promote collaborative research. COAST supports the reinforcement and expansion of the networks established through XLab Day. COAST, the organizer of XLab Day, stands as a beacon for Korea's science and technology society. Established in 1966, its mission has been to safeguard and amplify the rights and interests of scientists and engineers. Our endeavors extend to enhancing the public's grasp of science and technology and furthering its development through comprehensive policy research, planning, surveys, and consultations. As Korea's largest science and technology network with over 600 affiliated organizations, we are committed to promoting domestic and international cooperation and exchange. In the transformative era of the 4th Industrial Revolution, where science and technology spearhead groundbreaking challenges, COAST remains dedicated to its role as a facilitator of scientific and technological innovations.
안녕하십니까. 한국과학기술단체 총연합회 회장 이태식입니다. 과총은 사회참여 확대와 권익신장을 도모하기 위해서 1966년 설립한 공익법인으로서 과학기술단체 육성 지원과 다양한 정책 연구, 기획, 조사, 자문을 통해 과학기술 발전에 기여하고 있습니다. 저는 2023년 과총회장으로 취임하여 새로운 도약을 위한 비전 프라이드 코스를 선언하였으며 이를 실현하기 위한 운영 목표와 추진 전략을 중심으로 빛나는 100년을 향해 모든 역량을 집중해 나가고 있습니다. 더 나아가 우리나라가 미래 핵심 기술 분야를 선도하는 기술 주권 국가로 자리매김하도록 국가적인 원팀 플레이를 이루는 데 있어 강력한 조력자가 되겠습니다. 대단히 감사합니다.
BrainLink XLab Day aims to broaden both domestic and international talent networks and become a global hub of talent. By making XLab Day into an event that scientists and engineers worldwide eagerly join, we intend to elevate Korea's global standing and bolster national competitiveness. Global Talent Hub, BrainLink XLab Day.
예, 감사합니다. 예, 그럼 오늘 훌륭한 강의 내용을 들려주실 연자분들을 소개시켜드리도록 하겠습니다. I would like to introduce each of the international speakers who will share the valuable insights with us today. And Professor Teruo Okano from University of Tokyo Women's Medical University, Tokyo, Japan. And would you mind to stand up and say hello? Thank you. And Professor John Rasco from Australia. He is from the Royal Prince Alfred Hospital, Sydney, Australia. Would you mind stand up? Yeah. And and Professor Pamela Yellick, at Tufts University, Massachusetts, United States. Thank you very much. And Professor Sang-Jin Lee, Wake Forest University, North Carolina, United States. Professor Mime Egami from University of Utah, Salt Lake, United States. Thank you very much. We have another Sang-Jin Lee, University of Hong Kong. Okay, thank you very much. And I'm sure that all of our speakers are very tired because of a long travel to come to Seoul. But I really appreciate your kindest time sharing and coming to Korea. All right, so now, I don't have to introduce myself. So anyway, so now we would like to have the time of the presentation and talk from the international speakers. All right, okay. So Professor Okano, would you like to come to the stage and So let's welcome the Professor Okano, who will present the topic is cell sheet tissue engineering for the tissue engineering and the regenerative medicine.
Thank you very much. Just like arrived at Seoul 2 hours ago, and I'm excited to discuss with you about tissue engineering. And I'm an inventor of cell sheet engineering. Cell manipulation, cell sheet preparation, cell sheet manipulation, cell sheet transplantation. That's a new technology to open up the treatment, engineering, tissue engineering therapy. And this time, I like to introduce my basic technology. And tomorrow, maybe I can talk about how to treat the patient. And so think about materials, a little bit different from medicine. Just surface. And if we have one material, one structure show one function. But if we can change the structure, we can change the function. We call it intelligent or smart materials. And I really like to utilize this technology to medicine. And after I got a PhD in engineering university, and moved to medical school. And more than 40 years, I spent time in the medical school to utilize this concept. And I utilize the temperature signals and change the structure and develop new cell transplantation. So as you know, cell is different from normal beads, polymeric beads or other beads. Always, engineers think there is some interaction between materials and cells. But once cell stick to the surface, cell recognize the many information from the surface and change the structure, change the morphology, and try to grow on the surface. this process is important to magnify cells culture. But biologists know only culture dish. Sometimes they put the collagen or some proteins. That's it. People don't know how to recover the cells. That's the point. And 1990, I invented this surface is effective to design a cell detachment. I utilize these molecules, isopropyl acrylamide, and this is a temperature responsible, more than 32 degree globule, and under 32 degree solubilized by hydration. And I made a very homogeneous coating of this material polymers, and the 37 hydrophobic, and under 32 degree very hydrophilic, wettable. Therefore, after we make cell culturized cells on the surface and just reduce temperature, we can recover like this cell seed with adhesive proteins. And if we utilize enzyme, normal biologists always put the enzyme, disrupt the adhesive protein as well as cell-cell junctions. Therefore, we cannot make like these tissues. So that's why I initiated autologous cell seed we utilize and treat the patient. And tomorrow maybe I can explain detail of our therapy. But today I like to focus on this surface. And this surface, first I put the monomer and irradiate electron beam and polymerize on the surface. And this is a very, to remove the monomer, we use the large amount of water, and we cannot design detailed structure of surface. But recently, we invented second generation. First, we prepared the brocopolymer, and like this hydrophobic NIPAM chain connect with hydrophobic butyl methacrylate, and just spin coat, and we can introduce the NIPAM at 37 hydrophobic, and under 32 degree become hydrophic. But we have this anchor chain. Therefore, polymer, this brocopolymer, never reach out from the surface. Okay? So we to regulate the homogeneity and the thickness of coating layer, we are utilize like this spin coating, and you can see nano level thickness we can control. This right side thickness and the polymer concentration, when we change the concentration, we can regulate nano level of thickness. And this surface, cell stick to the surface, almost similar normal culture dish, and the reduced temperature, reduced temperature, cells start to detach from surface because water penetrates into interface between cell and the materials. And we can make like this cell seed. And mesenchymal stem cells myoblast, fibroblast, cardiomyocyte, cancer cells, and many cell line and primary cells. We can make cell seed by using regulated, controlled surface. And this is an example of after we remove the cell seed, we introduce the fluorescence molecule at the end of molecule. They never detach. After even we detach the cell, still polymer stay on the surface. And now university and one company and this is I made cystic cell seed tissue engineering regenerative medicine initiative, and company and the university, and we have succeeded to commercialize this culture dish. And this is one of basic technology to prepare the cell seed, because we cannot transplant cells nicely. Almost after we treat by enzyme disrupt important membrane protein. Therefore, even we can magnify the cells, but very difficult to transplant to the patient. So tomorrow maybe I can discuss effect of this cell seed and how to transplant and how to treat the patient. Thank you very much.
Thank you very much, Professor Ogano. I'm sure that you have many questions, but we would like to have a Q&A session after all the speakers are finishing the lectures. And now I would like to introduce Professor John Roscoe from Australia. He has another topic of there is now a path to treating thousands of previously overlooked rare diseases. So this will be another great talk on today. So are you ready?
Uh-huh.
Yeah.
Thank you, everybody, and it's such an honor to be speaking to such a wonderful group of individuals. I'm deeply honored to be here in Seoul, in South Korea, amongst friends, and I am looking forward to hearing all of my colleagues. I've prepared a little hors d'oeuvre today to introduce what we'll be talking about in the coming days, but in particular to highlight gene therapy, which is an area I've been passionate about for 3 decades. And as you can see, I have tried to practice some Korean in introducing this topic. Forgive my pronunciation, but I read this as Gujin Gamne. Gujin Gamne. And the reason for practicing my Korean is because yesterday I had the opportunity to visit Haejun's museum very close by here. And I reflected on Gujin Gamne, which means to me, for those of you who don't speak Korean like myself, it means sweetness can follow bitterness as a practical, literal translation, or after suffering can arise pleasantness, or in more familiar translation for English people, maybe every cloud has a silver lining. I think that is a familiar translation, but probably the most modern translation would be no pain, no gain. So if that means any resonance to anybody here, it means in relation to gene therapy that I think, as we were taught some 450 years ago by the great scholar Haejun, who wrote the encyclopedia the greatest encyclopedia of medicine in Korea 400 plus years ago, he taught us that perhaps there is some optimism and hope from the suffering that, in this case, genetic or rare diseases brings. Maybe we can bring some sweetness to the lives of those suffering. We stand on the shoulders of great pioneers, and just like Heo-jun, these are two great pioneers that we also need to reflect on, who passed away within 2 years of each other. On the left is the priest Gregor Mendel, who taught us about the principles of inheritance, and of course Sir Charles Darwin on the right, who taught us natural selection. And it is on those shoulders that we now can make the jump after 150 years to molecular genetics and the understanding of how genetic information is transferred within populations and also from one generation to the next. And it is that technology, which we now call biotechnology or molecular biology, that I wish to highlight today. And so to this audience, I really don't need to be too basic, but I need to remind you that the great German pathologist Rudolf Virchow taught us that simply one cell gives rise to two. He spoke in Latin. He wrote that omnis cellula e cellulae, meaning one cell gives rise to two. And that is such a simple idea, but it is the idea that if one gives rise to two, 2 to 4, 8, 16, 32, 64. And that concept means that we have a tool at our disposal, as we've just heard from Professor Okano, to use cells as a vehicle to treat potentially genetic disease, because we now understand that within the nucleus of each cell there is genetic information which is passed down through the generations. And we ask the question, if that genetic information has got a disease-carrying capacity that can be passed down and inherited, why not? Can we not modify or alter the genetic information in cells to achieve a cure or a therapeutic benefit? That is a simple and straightforward principle which has caused me to work for the last 3 or more decades in order to try and solve the technical problems of achieving this goal. So why do we want to achieve this area of gene therapy? Well, it's because many genetic diseases exist in humankind, which I'll elaborate on in just a moment. And so this technology is capable of targeting the root cause. If you are able to correct the root cause of a genetic disease, you may have a treatment that is available for the life of an individual person, although at the moment we do not cross the germline barrier, which is to say we do not cross the possibility of using inherited genetic information that is currently prevented in all countries over the planet and has not yet been overcome, except one example in China that many of you would be aware of. And so it may be the only option available to those people suffering from rare genetic diseases, and ultimately the goal is to replace a genetic abnormality with a correct version of the gene, or to correct that gene. I used to call myself a hematologist, and I still do practice as a hematologist, but now I think more of myself as a rare disease physician. And the reason is shown here, because rare diseases, even though by definition they are rare, collectively, if you add them all up together, they are actually as common as the burden of disease as the most common diseases altogether. So this graph here is simply from the USA, but it simply shows you that if you add up all the rare diseases that affect humans, it's as common as diseases like diabetes, major public health issues, but we don't spend anything like the same amount of money in rare diseases as we do in these common diseases like diabetes or cancers. How can we explain this information? Well, it is summarized here. Currently, based on the collection of diseases that affect humankind, just like it began with Heo-jun, the Encyclopedia of Diseases, we now know that there are some 7,000 diseases that affect humanity, of which 80% have a genetic cause as their basis. And so these people with so many thousands of genetic diseases are denied any form of specific therapy. Indeed, 95% of individuals have no therapy other than symptomatic or potentially assistive therapy. So this is an incredible unmet human medical need that we need to address, and I hope to convince you in the remainder of the talk that we are starting the era of genetic technologies and genetic therapies right now. There are two ways that we can introduce genetic information into cells. On the left-hand side is the in vivo approach, where we inject the genes into cells through the circulation or through the CSF or intratumorally into a cancer. On the right-hand side, we take the cells out of a person's body, and then we genetically modify them outside of the person, and then we can either freeze them down and store them or immediately return them fresh to that individual person. Genes are just chemicals, and they are comprising some 3 billion bases in our nucleus of our cells, and those chemicals they are basically an instruction set that tells the cell what to do. They need to be read by an intelligent system, and that intelligent system is present in all of our cells. So the trajectory of gene therapy is always going to be cellular, and that's why I highlight cells from the very beginning. On the left, the trajectory is through the circulation typically, or directly into the cells. On the right, it is outside of the body. Nevertheless, the destination is the same. And we must always embrace the use of clinical trials and evidence, empiricism in developing these technologies as the fundamental cornerstone for progress in cell and gene therapies. So look at this example now. This is like a running show of all of the 3 billion bases. And we can run down the list and say, what disease is a person going to be affected by an error in 3 billion bases? Well, the first example I will give you is an error in the DNA causing cancer. Because over the last 10 or 15 years, we have been working towards the use of immune cells that are modified with industry, and this is an example of these CAR T cells, the genetically modified or reprogrammed T cells that lead to a treatment for many blood cancers, and hopefully we will expand those to treating solid tumours in the coming years. This treatment is revolutionary. At my hospital, we've treated over 130 individuals, but there are tens of thousands of individuals now with commercially approved CAR T-cell therapies. This is one of the first human beings on the planet who ever received CAR T-cells. Over a decade ago, this is Emily Whitehead, who now is part of the Emily Whitehead Foundation with her parents, and she was at death's door from leukemia. She was not being able to be treated by any chemotherapy. It had failed. Bone marrow transplant had failed. And in UPenn in Philadelphia, she received the first ever, within the first 3 people, CAR T-cell therapies. And here she is smiling with her little dog, 5 years free of cancer. Very smiling. She had a single injection of her own reprogrammed cells, and she was completely cured of this life-threatening disease. And her physician said she would be dead within a couple of weeks. And now she's a smiling young lady. In fact, it's now the case that she's more than 10 years post a single injection of these reprogrammed cells that led to the eradication of her leukemia. And you can see her dog is not looking as well as she is after so many years. One is getting much older than the other one. Let's look at another quick example, because I know we only have a little time. And so the second example I will give you very quickly is thalassemia, a disease that is much more common in human genetics. The gene is carried by 5% of the world population, and it leads to anemia and chronic weakness and tiredness, and many complications due to the consequence of recurrent blood transfusions, which allow these individuals to survive. I love this photograph because it's the last image that I was able to take of a patient of mine. He allowed his image to be shown. This is Jack, and this is the very last blood transfusion that he ever needed to receive after he received ex vivo gene therapy with a technology developed by a company that we worked closely with over the last decade called Bluebird Bio. And after this treatment, he never needed to receive any more transfusions for the rest of his life. And he's now 8 years post Bluebird Bio's approved therapy for beta thalassemia, and he's no longer transfusion dependent, indeed doesn't need any transfusions. The last example that I will quickly give you is one I have been passionate about for over 3 decades, which is the bleeding disorder haemophilia, divided into 2 main types, haemophilia A and haemophilia B. And this treatment has now revolutionized the treatment of the therapies available to people suffering from this long-standing, long-recognized genetic disorder, perhaps the oldest known inherited disorder of humans ever, described in texts from the Bible some 2,000 years ago. And now this is approved in the USA, in Europe, and expanding the approval for this therapy in many different countries, and I will elaborate on that in the coming days. So let me close and bring this to a finish by simply reflecting on the fact that the technology is now proven. We can reliably and safely introduce new genetic material into human cells and achieve a therapeutic benefit. In the case of haemophilia, we have follow-up for many more than 5 and at times 10 years, and it is showing no evidence, at least in the case of haemophilia B, of any reduction. And yet society is very slow to consider the implications of this technology. There is a great deal of ethical and moral responsibility. The lawyers, the government, many of the reimbursers and many individuals, the philosophers, have not kept up with this technology. The technology has rushed ahead of where we are as compassionate, moral, ethical human beings. And we need, I think, to make the public more aware of not just the science, which we are all passionate about, but also some of the implications for life and a compassionate life, and bringing a future of sweetness to the otherwise bitter lives of those suffering from rare diseases. The last concept I will leave you with now is that the whole genome is druggable. Using new technologies beyond the simple approach of adding genes, we can now edit genes using the Nobel Prize-winning technologies of CRISPR-Cas9, and that means now that all the genome is druggable. We can modify the genome at our will, add, subtract, and modify, and this brings real opportunities to a future benefit. Thank you very much. 감사합니다.
Thank you very much, Professor Roscoe. It's from the viewpoint of another orthopedic surgeon. It's very great. We have another rare disease in the orthopedic field is spinal muscular atrophy. That is the new drug really helping the children and parents. So thank you very much. And now we would like to move to the next talk. Professor Pamela Jellick from Tufts University. She has a topic, how genetics can guide strategies for the tissue engineering and the regenerative medicine. Right?
Excuse me, I was just asking you about the pointer. But very nice to see you all, and thank you so much for this amazing opportunity to come to visit Seoul and Korea again. I'm looking forward to all 4 days of this event, which is so very exciting. And it was just lovely to actually follow John, because he gave a lot of the details about some of the genetics that I'm not going to go into detail about. But I'm going to talk about some other approaches that are based on genetics to help us treat disease and also apply them to tissue engineering and regenerative medicine and dentistry. Okay, so let me see if I can do this. Okay, good. So I'm going to introduce you guys to a model that you may not be familiar with, a genetic model, which is the zebrafish. Have any of you heard about the zebrafish? All right, well, that's good. You're going to learn something new. Sangjin has. Very good. It's a very, very, very popular model, but it's not a mammal, and so you usually need to move up from the zebrafish to a mammalian model and then to humans, but it has a lot of strengths, which I'll tell you about. And kind of in my laboratory, I first got introduced to the zebrafish as a developmental model, and then later got into tissue engineering and regenerative medicine. And I really love the way the two fields kind of interplay with one another, and I hope you agree with me by the end of this talk. So the zebrafish are this small vertebrate, so you can't see my pointer, but You can't see either pointer. But if you look at this fish on the right-hand side, it's about 18 hours post-fertilization. And I hope you can appreciate, you can already see the eye developing. You can see somites going down along the bottom, and kind of underneath and to the right of the eye is the heart that's developing. So it has very rapid development. The yolk is in the middle that gradually gets resorbed as the fish grows. And because the fish are external, you can watch them from the single cell stage. They divide synchronously every 20 minutes, so you have a clutch of about 200 embryos, as opposed to mice, which have maybe 10 or so, if you're lucky, 8, less than that. But you can watch them from the single cell stage. If you keep them at the same temperature, they'll develop synchronously. They're free-feeding, free-swimming by 3 days post-fertilization. So at that point in time, they have all of their jaws. They have teeth that are developing. They're sexually mature about 2 to 3 months, depending on how well you feed them, which is a little more rapid than mice. They're transparent, so you can use a lot of transgenic reporter fluorescent lines to show up the tissues that you're looking for. If you're looking at heart development or skeletal and cartilage, muscle, bone tissue development, which is what we work on. But most importantly, the genomes are very similar between the zebrafish and humans, believe it or not. They're about 2 times 10 to the 9th base pairs haploid genome, and the organization of the genes on the chromosomes, which is called synteny, and the nucleotide identity, the amino acid sequence identity is similar, which means that the functions are similar, which means that you can study gene mutations in the zebrafish and learn about human diseases. You can also express human gene mutations in the fish and study the molecular consequences and learn from them to apply to humans. So I'm going to talk briefly genetics. Everybody has to roll their eyes because genetics can be very overwhelming. But this is a forward genetic mutagenesis approach and screen. So just think about it this way. Reverse genetics is when you say, I want to knock out this gene and see what happens. Forward genetics is when you randomly mutagenize a developing male zebrafish by introducing a chemical mutagen, bathing them in a chemical mutagen that on average induces a single base pair mutation in each of the developing sperm cells, right? The fish continuously produce germ cells. It has to be a dividing cell for the mutagen to get in. So you can take that, and I don't have a pointer, but if you look at that red arrow, that's your chemical mutagen. You treat that male a few times with this titrated amount of mutagen, cross it to a wild-type female, and breed these green M's that are a collection of F1 fish that all have unique point mutations. So 1 base pair in 10 to the 9th base pairs. Then you raise those to sexual maturity, another 2 to 3 months, cross them to another wild-type fish, and end up with these F2 generations, all these individual F2 generations, of which half of them share a single base pair mutation in a heterozygous, so only in one, they're not homozygous, they don't have 2 mutations, they only have 1, and the other half are wild-type, which means that every time you pull a fish out of the tank, you have a 1 out of 2 chance, getting away from the microphone, of pulling out one of these plus M's. So 1 out of 2, 1 out of 4 of them that you set out on average is going to be 2 plus minuses or plus M's, among which 25% are going to be homozygous recessive and they might show a phenotype. They're going to show Mendelian inherited homozygous phenotype. And so we did this sort of a screen, and this isn't my idea. This is very popular and was done large scales, both in Tübingen and MS General. And what we were looking for, though, was craniofacial anomalies. We were looking for craniofacial and skeletal anomalies like these, for which we had some gene mutations that are known, but many which were not. And so we screened our fish for those kinds of defects. So most importantly, we were looking for the vital ones. A lot of the mutations were going to be early lethals, which meant, for example, if you look on the left side, all of those guys have very abnormal heads, which means their pharyngeal arches are malformed. And you can look at the blue stained cartilages, and you can see how different they are. And that means they can't feed well, they don't float as well, they don't form their, they're just sick and they die. But they're still very interesting in that that gene is required for normal craniofacial development, right? When you have that single base pair mutation, it disrupts that gene that is required for normal craniofacial development. And then these ones down at the bottom, those are the adult viable mutations. And I hope you can appreciate the midface hypoplasia in the bottom with the arrow, and those also had some skeletal defects. And so what you can do once you have these fish that have these phenotypes, you can collect tail clip DNAs from their normal siblings and from the affected siblings. And then it's easy. You can do whole genome sequencing, find out the area that differs between the two, identify the gene mutation, and boom, you have a new gene that could be a candidate for some of the human orphan diseases. And that's exactly what we did. Okay, I know we don't have a ton of time, so what I'm going to move on to is talk about how you can make humanized zebrafish models for human diseases and rare diseases. So here's another rare disease. This is 1 in 2 million people that have a disease called fibrodysplasia ossificans progressiva, or FOP, which is a disease that's characterized by this progressive heterotopic ossification of soft tissue. So you can see these skeletons just get more and more deformed, and eventually they die they can't breathe any longer, and you can't operate on these people either because any sort of an injury induces this explosive heterotopic bone formation. So it's caused by mutations in this gene, a receptor called ACVR1. The most common is this R206H, arginine 206 histidine mutation. That's the most common. And that is the first gene that we identified in the zebrafish as far as the zebrafish ortholog for this receptor family. And that was just happenstance. It had no affiliation with FOP at that time. But it's involved with upregulated bone morphogenetic protein signaling, which makes sense. It's a heterotopic ossification. It also has neofunction by activin A, which is a whole new thing. It's progressive, very painful, debilitating, and fatal. And there's no cure. There's some palliative treatments for it that can make the patient feel a little bit better, but there's no gene therapy cure, for example, or there's no way of treating it right now. But what we did is we created zebrafish that are expressing this human R206H ACVR1 gene mutation, and we showed, I hope you can appreciate, that we can see the second skeleton forming up here. It's just running parallel to the other skeleton. We're seeing vertebral fusions. We're seeing, thank you very much, a lot of other things that, all right, now I'm in trouble. You're in trouble. No. And you can see this heterotopic bone formation. All the white is bone, right? So great, we've got a model. So we can study what happens downstream now, and we can also study what happens with normal heterotopic ossification that isn't genetic necessarily. And then we can use the fish to screen for treatments. These fish are really tiny. They're a millimeter in diameter, so you can put them in 384 well plates and screen for small molecules. But you could also, if we could figure out how to exploit these explosive bone signaling pathways to regenerate bone when we want to, in a controlled manner though, right? You don't want heterotopic ossification happening. And so how do you do that? One of the things we did, and this is a publication with a wonderful team at UCSF, is we used the zebrafish as a platform. This is a little bit of a complicated slide, but just let me tell you that there were some patients who seemed resistant to the progression of the disease. They had the R206H mutation, but they didn't succumb to it in the time that most people do. And so the doctors, physicians were wondering whether they had compensatory mutations in them that were offsetting the FOP signaling pathways. And so they did whole exome sequencing of these patients, which is an ultra rare population now, right? It's already an ultra rare population. and identified some genes that were in the BMP signaling pathway, and those are shown here. So this BMP R1A, R443C, and this ACVR2A, V173I. And so my colleagues at UCSF characterized those genes in vitro models of FOP. So they had FOP cells that they were growing, iPSCs, that they were characterizing with these signaling pathways. And then we had the fish. And another advantage of the zebrafish is that we didn't have to wait for heterotopic bone to form, because BMPs also modulate dorsal ventral patterning early in the embryo, like by 48 hours, 24 hours. And so what we could do is take our FOP fish, microinject these mRNAs for the BMP-R1A and ACVR2A, and then look to see what happens by 24 and 48 hours. Does it ventralize or not? And unfortunately, what we found is that neither in the zebrafish nor in vitro did those gene mutations compensate for FOP. They didn't block BMP signaling. But at least it established the fish as a platform that could be used for other, whoops, I think I did that, for other diseases. Okay, then moving on, what else are we doing with these FOP cells? Well, we can get teeth from patients that have FOP. They exfoliate their teeth the way normal people do. Sometimes they have adult teeth removed for orthopedic reasons or impacted wisdom teeth, whatever. And so we get those cells when they can send them to us in time that we can get any viable cells out of the dental pulp. And we can expand them in culture to tens of millions of cells, freeze them down, and then use them for our analyses. And one of the things that we wanted to do, well, what you can do, just although in vitro culture is very simple, it avoids some of the confounding factors that you can have in patients when you're analyzing their serum. Perhaps they're taking other medications, or they have underlying diseases, or their diets are different, or their exercise levels are different, their climate is different. And so it's very simple, but you can perform metabolic profiling on these cells, and I'll talk a little bit about that. You can look at the secreted exosomal vesicles from these cells and how they differ from normal dental pulp stem cells. And then you can study their in vitro differentiation and hopefully use them for tissue engineering regenerative medicine strategies. And so what are metabolites? So for those of you who haven't characterized it, they're really everything, right? They're metabolites and metabolic pathway intermediates are made when the body breaks down food, drugs, chemicals, or tissues. So it's everything along the way. Small molecules that supply the cell with energy. They really do everything. Their functions are fuel, structure, signaling, catalytic activity. They're really very, so it's a big group of very important molecules. So what we did is we took these FOP dental pulp stem cells and normal dental pulp stem cells. We cultured them, and we did metabolic analysis of them. And we found 100, this is just a normal media, not differentiation media, so not trying to differentiate them into bone-forming cells. We found that there were 183 cellular metabolites that were significantly differentially expressed. 179 of them were twofold. And what we're hoping is that we can find metabolites that might be indicative of a flare-up that might be coming for these patients, because then you could give them the medications that have other serious side effects, and you don't want to have them be taking that all the time. But if you could do that, that would be a wonderful tool. And it might even be able to develop more effective therapies for treating people with FOP. And you could certainly manipulate these signaling pathways in the zebrafish using these whole genome editing methods that there are, that were just described earlier by Dr. Rasko, and see what might be the most appropriate targets for eventually moving up to human, mammalian models in humans. And then in the last few slides, I'm just going to talk to you a little bit about exosomal vesicles. This is a really fun project that we just started a few months, maybe almost a year ago, not even, with a very talented master's student in the lab at the dental school. And for those of you who aren't familiar with EVs, they're these cell-derived nanovesicles that are fabricated inside of the cell, they're packaged up, and they're released, exosomes, and then they're taken up by another cell in a targeted manner. So the surface of the exosomes enables them to be recognized by the cell that they're supposed to be getting to, and it picks them up. And then they deliver whatever their cargo is inside, which are microRNAs, DNA, RNA, proteins, et cetera, et cetera. And because EVs are very important, they're being considered as a cell-free therapeutic method that might have great potential because it doesn't involve cells. You don't have to ex vivo, you don't have to culture cells and then introduce them back into the person, which the FDA is not thrilled about, right? Because you expose it to serum and all these other tissue culture reagents that might have contaminants in them. I mean, it's a very difficult process to get an FDA-approved drug that includes cell therapies, but that includes cells. But if you could create EVs or even better understand what are the EVs in the cell, these different cell types that are the most important, you could find a very targeted delivery method that would be very focused. Again, as I mentioned, they're only taken up by certain cell types, so you could design them to be taken up by your cell of interest. So you could design them to be taken up by soft tissues to prevent them from ossifying, heterotopic ossification from happening, for example. And so what we're doing in the lab right now is we're taking normal dental pulp stem cells, normal periodontal ligament cells, and then our FOP dental pulp stem cells, and growing them up in large T175 flasks, collecting the media. Well, you move it into serum-free media that doesn't have any exosomes in it, and that to confound your data, collect them, and then go through this. We're using a series of filtration columns to get these little EVs. Ultracentrifugation is another way. And those little bright dots down there in the bottom are what you can see in the Zeta View particle tracker to know that you've got your exosomes. And then what next? Well, then what you can do is see your profile so you know that the EVs are about the right size. And then you can get them from saliva is another nice method, of a non-invasive method to get them from. And then really what you want to do, though, is to know what's inside. And some of the important components of the cargo are microRNAs, and there are also proteins, mRNAs, and lipids. And for those of you who don't know what microRNAs are, they're an evolutionarily concerned, very small, 22 base pair regulatory RNAs that participate in all cellular processes. And they control gene expression by binding to the 3' end of a messenger RNA and blocking translation and targeting it for degradation. And so they mediate all sorts of pathways. And this is just kind of a schematic of what it is. DNA, mRNA, messenger RNA at the top that turns into a protein. If you have your microRNA, it will bind to your mRNA and block translation, and you'll lose that. And the other important thing is that microRNAs will target a whole multitude of mRNAs, so you can block all sorts of signaling pathways, related signaling pathways, at the same time. And so we're interrogating our EVs from our in vitro cultured dental cells in this way. So on the very left, it's very tiny, I apologize, but you can see those are our mRNAs, our miRNAs, very small miRNAs. And then we're using those to make cDNA libraries, which then we will sequence. So we're right at the important point. We know that we've got EVs that we can make enough miRNAs out of, and we can make libraries from it too. So all we have to do now is complete the set of our 3 different cell lines of each cell type, 3 replicates each of those 3, sequence it all in one sequencing lane. That's $1,800. But then we'll get all of our sequence out, and then we'll have our work cut out for us trying to figure out what in the world is going on and what important differences or commonalities are there. So in conclusion, and as was already talked about in our first talk, genetics are a very powerful tool for interrogating signaling pathways in human health and disease. And the zebrafish are another model to add to our our repertoire of tools to use. The zebrafish screening platforms could be very valuable for identifying small molecules. Cell-free therapeutic strategies might be a better way to go if you're trying to get a product approved by the FDA or whatever regulatory system you want to get it into people. Simpler, more basic, but maybe even more targeted and effective. Metabolomics profiling is really interesting. We're really looking forward to working with that. And then microRNA profiling, too, can lead to just better, hopefully, new insight into regulatory mechanisms for common and rare diseases. And these are some of the people in the lab or in the lab now. Nothing would get done without all the people that are working hard in the lab while I'm traveling around, and our very generous support. For a second, I'm just going to put on my International Association of Dental Research hat. I'm the president-elect of the IADR, which is a wonderful organization that is not only dental, oral, and craniofacial, but it has lots of wonderful things. Our meeting in June of 2025 is going to be in Barcelona. We've missed the proposal submission deadline, but the rest of the deadlines are still open. If you have any desire to go, I strongly encourage you to do so. Thank you very much for your time.
Thank you very much, Professor Yeli. And I'm one of the audience that I can, touch with about the, FOP. I have one patient FOP near my city, and whenever, the parents call me, that is an emergency. I need to use the prednisolone, to have prophylaxis of flare-up. So hope that we can have another, advance, to treat these patients. Yeah. It's just 5, 5 years old. Yeah. Yeah. Yeah, we will talk about another 3 days until Saturday. Thank you very much. All right, so now we move on to the next speaker, Professor Sang-Jin Lee from the Wake Forest Institute for the Regenerative Medicine. He has a topic of 3D bioprinting, bringing your imagination to life. Are you ready? Okay.
Yes, I truly appreciate organizers for giving me this opportunity to share my insight on bioprinting in regenerative medicine applications. So I'd like to start my presentation with this movie clip. That is one of the famous movie, Avengers series, that was released in 2015.
It's Ultron's-based consciousness. Informational noise. Soon.
How soon? I'm not being pushy.
In the
We're
movie,
imprinting a physical brain.
Korean
no
scientist Helen Jo,
okay?
she utilizing 3D printing technology
There's
to
no
creating
possibility of
tissues.
deterioration. The nanomolecular functionality is instantaneous. The cells don't know they're bonding with simulacrum.
She's creating
And particularly, last scene shows that skin printing. So now the imagination is turning into reality. So the team at the Wake Forest developed inside skin printers, bioprinter, that composed of 2 major components: 3D scanner that can detect skin wound, and dispensing module that can deliver skin cell directly into skin wound regions for treatment. And this has been validated in full thickness skin wound in pig. And as you see here, utilizing autologous cell, we could find that very active tissue regenerations. And so why 3D printing in the field? So it is basically layer by layer process. So we can fabricate such a complex construct relatively easily compared to other fabrication methodologies. So 3D printing impacts many industries, including fashion, automobiles, and architecture, and food, as well as biomedical field. So in 2010, The Economist published this illustration showing a machine that print organs in the operating room. And since then, number of publications related to 3D printing in format dramatically increased, as you see here. And now we can find many promising outcomes from many laboratories. So what's the 3D bioprinting? So unlike conventional 3D printing, it requires many components start from medical imaging to obtain a patient tissue structure and generating a 3D CAD model for a 3D printing process. And hardware bioprinter, currently there are many printing modalities utilized for bioprinting purpose. And biomaterial as a bioink that provide proper printability as well as microenvironment to accommodate tissue-specific cell types. And lastly, translational cell source is major component for cell-based bioprinting applications. But most importantly, 3D bioprinting aims to develop and provide personalized bioengineered tissue construct for improved patient care. So to achieve this goal, especially our group, WFORM developed integrate tissue and organ printing system that is designed to fabricate a complex tissue structure by sequentially printing biodegradable polymers and cell-laden hydrogels, creating human-scaled structurally stable tissue construct. So I'd like to show how ITAR works. So the shape of tissue is ensured by representing medical imaging as a 3D CAD model and translating this model into a pattern that controls printing motions. So as you see here, we target to print that human-scaled ear cartilages. So using three major components, we have cell-laden hydrogel that contain chondrocyte, and we have biodegradable polymer, polycaprolactone that support structure. But at the same time, we have sacrificial material that support printing process to build 3D structures. So after printing, you could find that human-scaled ear cartilage structure that containing live chondrocyte. So in order to make sure that this could be mature, so when we implanted this construct in a mouse subcutaneously, we could find that there are cartilage-like formation as well as mechanically similar like ear what we have. And we can also utilizing this technology to build a bone scaffold, so that could exactly fit defects, bony defect in the mandible we create. So depending on your design strategy, we could have effective bone regeneration, as you see here. And also we could utilizing this technology to build meniscus. So meniscus especially request highly elastic properties. So in order to have this capability, we use that silk-based bioink system to have a highly elastic property as you see here. But at the same time, this hybrid scaffold, hybrid bioink could accommodate meniscus cells to initiate meniscus tissue formation as you see here. And more importantly, we could have collagen fiber alignment that is very similar like native meniscus tissues. And cellular orientation is very essential for when you target to skeletal muscles. So using 3D bioprinting technology, we could have very organized myofiber structure. You just need a print skeletal muscle cell with one direction, then you can achieve highly organized skeletal muscle construct, you can see here. And when you apply this bioprinted skeletal muscle in muscle defect in rat, we could maintain their muscle volume, but more importantly, we could restore muscle function as you see here. And histologically, we can find that newly formed muscles in the defected region. And also, we could see bioprinted construct that is able to integrate with host nerve system and vasculatures. And a similar strategy, we could fabricate cardiac tissue with some such organization. So you need to print that cardiomyocyte in one direction, that we can have very organized cardiac tissues, as you see here. And during that culture, they start beating automatically. And when we treated with a known cardiac drug, they respond to correctly. And creating composite tissue is quite challenging because, for example, muscle tendon unit, because you need a very specialized interfacial tissues. So using 3D printing, you printed muscles and tendon, and then you can creating interfacial tissue that is muscular tendon junction. You could have between the tissue, and same strategy we can composite osteochondral that is bone and cartilage, but we can also creating functional interfacial that is calcified chondrogenic region between bone and cartilages. So this is kind of summary. So we could see the valuable progression on single tissue like bone and cartilage and muscles, and hollow tissue with multicellular cell, and tissue organization, and like skeletal muscle and cardiac muscle, and composite tissue like osteochondral and muscle tendon unit. But ultimately, we could target to print more solid functional organ like kidney and heart and liver, but that requires very complex and very small micron scale functional unit like vasculature and nephron in the kidneys. So in order to achieve these goals, we need better isolation of multiple cell source, and we may need multicellular and multimaterial printing with high resolutions. So once we establish all the necessary components to build a clinically applicable 3D bioprinting workflow, we may be able to bring this technology to life in the clinical side. And thank you for my, especially internal and outside collaborator and my team member, as well as our funding agency to support our research. Thank you.
Thank you very much, Professor Lee. And I'm sure that if you have more and more this work, our role of orthopedic surgeon will disappear. But I'm thank you very much. And now we move on to the next speaker, Professor Mime Igami from the University of Utah. And she has a topic of how to realize the second generation cell sheet RM for patients. Thank you.
Thank you very much for your introduction. My talk is designed to reflecting the title of this talk concert, the strategy to secure core technology to develop advanced regenerative medicine therapies. The secure means various meaning that to continue and really realize the advanced regenerative medicine is not easy. And we need, and we are living in a very practical world. So I'd like to share my thought with you for the discussion. So I'm partly University of Utah, but at the same time, together with Professor Okano, we established cell sheet tissue engineering regenerative medicine initiative in Japan, not only for Tokyo Women's Medical University, but to more accelerate international network and also commercialization of this core technology to the real world. And currently, I am a part of some Italian effort as well. So because I don't know today's audience how much you are familiar with the regenerative medicine tissue engineering, this cell sheet engineering technology is let's say that this is more like a material-free type of tissue engineering method compared to more material-based tissue engineering. So of course, not only cell sheet engineering technology, but also a spheroid-based tissue engineering or 3D printer level or a mesh mode to make structure of the cell. And then take out these like a metal needle or mesh mold before transplantation is another option as a science and technology. So the Professor Okano already talked to you, and also he will explain more details tomorrow. But just 1 second. So this cell shielding technology is developed by Professor Teru Okano, and this is quite a simple way that some responsive culture dish to harvest cells in sheet form without damaging extracellular matrix or cell-cell junction, just by lowering temperature of the culture dish. And 2000, the one venture company was established, and this company started to sell some responsive culture dish. So the up cell or rep cell or hydro cells to support almost like a first generation cell sheet-based regenerative medicine effort. However, that the upsells or others are more focused to help first generation regenerative medicine, like more endothelial cells, whatever. So that now we are shifting to more allogenic cell, iPS cell, that we need more the custom-made, fine-tuned surface is required to help the researchers' effort. And as I said, the researchers are really challenging to culture a wide variety of cells to achieve effective cells culture for regenerative medicine. And to do that, we need, if the culture this side is not so flexible, the researchers started to do the more pre-coat way or the different methods to cell culture, which makes it a little difficult if you want to shift to mass production by company. It is good for scientists, but it is not so good for the company. So, and also when I met the physicians of even like IPS Cell Research Institute, others, they mentioned that there are various new type of cell harvesting method is coming as science, but as physicians, they prefer some responsive dish because they don't have to worry about any additional risk for the patient. So I really recognize that some responsive culture surface is necessary not only for first generation regenerative medicine effort, but also the second generation or different type of cells. So this CS term decided and launched this new type of some responsive culture surface, so-called smart surface culture ware, actually this month to the market. And I really appreciate not only own small network members in Japan, but also the friends in Korea, like Professor Brian Che of Inha University, or Dr. In Kyun Shim of Asa Medical Center, Professor Kyung-Soo Kim. They really help to evaluate this surface to how to improve, how to optimize the surface to culture various different type of cells in smooth, stable way. And during the time that following the Professor Okano's effort, the Tokyo Women's Medical University's team drive more of the clinical application, but at the same time of the tissue engineering by using this cell sheet technology. Some people said that cell sheet is two-dimensional, but to add more technologies, the Tokyo Medical University is trying to make more tissue engineering 3D structure by this 2D type of cell sheet technologies, and try to collaborate more wider with other universities. sorry. As product, the two products, for cornea was approved, and Japan Tissue Engineering Company is now is selling in the market, and also heart sheet was approved by accelerated approval in 2014 for cardiomyocyte for cardiomyopathy patient. However, that it was not fully approved this year. As of knee cartridge, it is autologous, advanced therapy at Tokai University Hospital, and also clinical trial of allogeneic cell is starting now in Japan. As you may know, that in Japan, that new accelerated approval system was introduced in 2014, and by now, 20 products are approved in Japan, and it is so-called regenerative therapy related product, but actually it's wide range of modality. So first part is the tissue transplant type of product is a left side, cell transplant type of product as well, ex vivo gene therapy product, and in vivo gene therapy products as well. And during the time that US approved more than 30 products, and Europe is more than 22, 3. So that though we had an accelerated approval, regulatory approval system, but still that up to 20 is our achievement. So that we have some discussion that whether this accelerated approval system is good enough, viable enough to really accelerate the core technology regenerative medicine to the market. So out of this cell sheet research members that currently that we are shifting not only patient own cell to allogeneic cell and also iPS cell to improve standard efficacy. And one company, Quorips, working together with Professor Sawa Osaka University and starting clinical trial, the physician-initiated clinical trial supported by the company is starting. And also that at the University of Utah, more complicated organs is challenged by utilizing codified allogeneic cells, such like knee cartridge therapy, kidney transplant, the uterus cesarean scar disorder management. That, as you know, that in Japan and Korea, the government restrict number of baby delivery by cesarean section only like two trial, and still the government is worrying that our birth rate is too low. So then, how to solve it is the issue. So this third project at University of Utah is not only for state of Utah, where the Mormon people are there, but also that it is for our needs in Asia as well. And at the same time that the Professor Bon Healy's company, NCH company, and Siesta work together, and by getting the great contribution by Professor Kyung Kim, that we are trying to develop in hemophilia treatment by CRISPR technology-based iPS-derived hepatocyte cell sheet, and we are really excited about it. So so key is that how to build a solid business model to treat mass number of patients by allogeneic cell sheet. The autologous the regenerative medicine has been tested in Japan, but still that autologous cells so varies by patients. But so core technology of standardizing or screening effective allogeneic cells is key, whether we use iPS cell or screening or develop screening technology of effective allogeneic cells to and develop master cell bank feasible and also that strong enough to support mass number of patients is the issue. And also that in Japan we are now discussing that proper integration of private treatment. If this private treatment is reviewed and approved by governmental level or committee, that still that data is very important to develop more solid regenerative medicine protocol and also science that which can achieve the full regulatory approval, and also that the venture company doesn't have enough money for such a stable and large production of cell products. So the cross-functional CDMO is a cell develop and manufacturing organization is essential. And also that this medical field is relatively domestic and very silo. So that to get the higher social interest, higher type of fund, and also more global sense of business, maybe that a wider scope have to utilize the cell culture technology to attract other industries, maybe another way to expand the business size. So because both healthy longer life and also like healthy stable food supply is a global challenge. It is a global issue, not local silo issue. So these type of concern is now getting together and also attracting the larger size of company like Nissin or Ham and Nikon to get together wider than before. So at Tokyo Mima University, Professor Shimizu and his team is trying to utilize this cell sheet engine technology for medicine to this cultured meat area as well. So and actually the new venture company of cultured meat decided to establish the first presence at this institute, and now they are moving to that Takeda's huge research center to again that expand their technology to the second generation. So this Integrity Culture, this is a Japanese venture company. They learn this stem cell technology or culture method at this the Institute of Tokyo Medical University. And they are moving to Shonan Science Park, which is a former Takeda R&D center of iPSL and others, and to expand their business development. And actually, the Korea is really leading in Asia to develop not only technology, but also regulatory framework and R&D free zone in Asia. So Japan still struggling how to regulate the effort to develop the new culture meat. We cannot eat the tested culture meat in Japan. So last month, this JCA, this is the Japanese Association of this culture agriculture means cultured meat. Really pointed out that Korea is moving in the right direction. Why Japan cannot do that? Always Japan and Korea watching each other and try to collaborate. And again, in Korea, that you have this new free zone, and I don't know why all men, no women are here, but still, these motivated people try to develop this area to again that very healthy and stable food supply for Korea. And not only for that, this is a Korean movie, the Chen Ji-hoon and Han Hyun-joo, very famous movie actor. That nice movie. We are watching it in Japan by special movie channel. That this is a cultured meat mission, people. But still, it is involved to like a government politics or fighting or shooting type of thing. But as a basement, they are trying to rescue the disease patient by leveraging technology for the regenerative medicine. So this type of movie really attract or tell people that these technologies are really collaborating each other, and something is happening to accelerate both the grand challenge in Korea. So that is very Korea is very smart to really grab that attention, and you have the KFRM to support the regenerative medicine research activities. This is what we don't have. We have Japan Science Technology or AMED allocate some of the budget to regenerative medicine research, but not specialized funding agency exists in Japan. So therefore, that moving forward to make us all happy, that I think that this cell sheet technology that we could bring to the we could achieve regulatory approval for several products, and this evaluation of treatment outcome data of this first generation outcome data and mechanism of first generation autologous product by second generation science and technology is very important. And the seamless improvement of cell engineering technology to the more three-dimensional or more challenging cells are very important. And also that cell sheet is very effective as test kit of other cells, quality of a cell or 3D culture. Therefore, I think that the collaboration is very essential. And let's use that some achievement of first generation to the second, third generation. So that I think that this X-Lab Day is a good opportunity that not only your own way of this strategy, but also please discuss with other strategic people and find some win-win structure. Actually, together with Professor James Yu or Sanjin Lee, we had discussion that how more material-based and non-material-based research can collaborate each other. But at that time, our both technology were not so advanced as now. But maybe we may try again in the future. So don't hesitate to communicate with other approach people. So thank you very much, and I really want to discuss with many people, especially young people, to discuss a good future. So this spiral, the expansion is a key for success, I think. Thank you very much.
Thank you very much, Professor Egami. So I am sure that we will have another, the fruitful discussion during the Q&A time. And all movies have a hero, and the hero comes the last moment. There is a Professor Sang-Jin Lee from Hong Kong, University of Hong Kong. He has a topic of advancements in the dental tissue regeneration, trends, impacts, and the future vision. So are you ready?
Yeah.
Okay.
Really gratefully, thanks for inviting me. I'm actually very honored about my printer's time today because I actually learned a lot of things from all of you. Because when I was a graduate student, and also I saw a lot of paper, actually proper kind of paper, cell sheet, because I just started my tissue engineering work from your research outcome and achievement. So today I just wanted to introduce the dental tissue engineering very roughly. Actually, I didn't bring my research topic, but everything is all connected to my research, and I just wanted to talk with you continuously first. And I just want to say it. What is a dental tissue engineering? In the body, there's a lot of tissue, and also in the oral area and craniofacial, these are all connected together. And also, we are saying this is a dental tissue engineering. Many people know that the dental tissue means that some teeth or some inside very specific things. However, in the dentistry area, we are saying all the some skin or some craniofacial bone, also inside some epithelium, even upper neck. So this is the oral area. So which means that these are very, very complicated, not simple. So more specifically, we can say the craniofacial bone and periodontal. Periodontal means that it's a teeth just hold it from some bone, and also there's a lot of layer by layer tissue, especially some gingiva and periodontal ligament. Also vasculature, there's some joint, and they all connected together. And also TMJ. This is also very complicated that it is connected with the bone and cartilage. Also, even though we have a lot of cartilage inside of the body, some joint area, this is kind of like one of a small joint. And also pulp dental. This is a teeth. Or maybe you guys have some experience of teeth decay and some pain. This is very, very painful, super painful, even couldn't get a sleep, right? So this is a teeth, and we are trying to recover these teeth based on the tissue engineering goal. Previously, there's no way to recover this pulp area joint with the materials. However, nowadays we are trying to transplant the stem cells into this pulp area. Let me show you more detail. And also oral mucosa. Many oral cancer occurred in the oral tongue and some of the mucosa area. So this is one of the big field that we are trying to remove the cancer tissue, then absolutely we need to recover it because the teeth is very thick. So if it is not simple, so if we remove the tongue, we should recover tongue using some biomaterials and stem cells combination. So this is the reason why I just wanted to say area. Today I would like to more focus on the dental pulp and dental teeth tooth regeneration. So what about the dental caries? Dental caries means that some young age, everybody has the issue dental caries. This is occurred from the biofilm. Biofilm, we can't see it. However, it is covered on the teeth, and this biofilm just trying to digging the dentin or some enamel. Then finally, we feel the pain. So how about this? If it is, we need to remove the distal biofilm and some bad things. Then finally, we need to recover this damaged area. So in the tissue engineering goal, we are using the hydrogel or some composite materials in order to cover this damaged area or recover this tissue. So more specifically, if damage is severe and serious, we should remove the dental pulp. Then finally, we need to replace it on another alternative solution, such like the MTA, this is commercial one, and also some composite or some liquid like this. Then finally, we just wanted to recover this damaged tissue as original from the damaged one. Then below the capillary, and there's a nerve, and we just wanted to say the fully recovery engineering based on the tissue engineering goal. So there's a lot of research. Previously, Prof. Pamela already described the dental pulp stem cells, and also in the oral area, we could get a lot of stem cell. Actually, this is originally, it is similar like our general mesenchymal stem cells, which means that it is multifunctional, can be bone, cartilage, nerve, or even vasculature. There's a lot of specific type. Then one of the biggest thing is we can get this from the wisdom teeth. Wisdom teeth means that this is a waste. So I'm in my hospital. I'm taking a lot of wisdom teeth from my student, and then they just provide me the dental teeth, and then obtaining the patient drive the dental stem cell or periodontal ligament, and there's many cells type. So this is a basic recovery. You can see the middle of upper. After making some defect, you manually this is everything is performed in the dental hospital and private dental clinic too. Then finally, we just replacing this empty area using the hydrogel with the stem cells. Actually, this is a stem cell approach is not allowed yet. However, this is a future trend, and currently we are investigating very much because the bottom area, piece of the bottom area, there's a lot of nerve and bone, all many tissues are connected. That's the reason why we are feeling the pain. Then final goal is generating the new tissue in the teeth. So actually, these pulp dead and we remove, but we just wanted to provide the new cells and some stimulation and new model based on our tissue engineering approach. So this is look at this. This is after recovery. This is research, not clinic. Research after that, we just transplant. Left pinky color is a dentin. This is actual teeth. But inside you can see the many cells ingrowth inside, and also could see the vasculature, right? So vasculature means that from the native host tissue is ingrowth inside the new transplant. Then finally we could see that there's some stem cells and vasculature, they all connected together, which means that we are investigating the many cell source and some biomolecule and exosome nanomicroparticle. Then final goal is making the new teeth from the dead teeth. And also the other technology, it is also 3D printed approach that we in our dentistry, we are also trying to making some living teeth using this 3D printing approach. Actually, this is very far from the clinical approach, clinical application. However, we are continuously checking how we can make the more living tissue, and also how we can provide the living things to the patient. So you can see that making some substrate using the polymer, then finally hydrogel and stem cell decorating all inside and outside and differentiate, then making some new tissue model. So also additionally, I just wanted to introduce about the new viewpoint that bioengineered tooth germ. Ethically, it is also far from the clinical area. However, this is this tooth germ obtained from the embryo, and then we just combine together. Then finally, making some dental tooth organ, and this is a very initial starting point. Then, what we can do after transplant in the body, actually we could see the new teeth formulation. So this is a very soft tissue. However, it becomes more sticky teeth like our tooth. So this is a more detail. So making the tooth germ and epithelial and mesenchymal and then combine together. In this case, cell sheet engineering also very potential. Also spheroid tissue engineering. Combining together, absolutely, if they do not meet together, there's not anything else happen. Absolutely, they should meet it. Then could they formulate it together? I don't know mechanism very well, but they just transform into the tooth very well. Then once many scientists trying making how to, so if so, they already checked the combination, but they just wanted to see the real teeth eruption and formulation. After making some germ organ, then transplant into the mandible cavity. Then scientists just found the right lower area, the tooth erupted. So this is a very fresh new viewpoint, and we are making how to solve some ethic issue or some immune issue. But this is just technology. We are still trying how to find the way. Then this is the final slide. This is a hot topic in the field, AI-based data running and data mining and machine learning, a lot of things. This is a recent publication in my institute, Hong Kong New Dentistry. And some scientists just established one mobile device, and then they took the image. Then this is the data actually. why we are doing this? A patient by themselves, they just took the image and then sending the photo, then we could see their shape and their some point, how they move. And also they just send the movie, then we could see their action, how this mouse or condition is okay or no. This is not very associated with our tissue regeneration cell function, but I just want to say this technology absolutely connected with our future tissue engineering goal, because this is a data and we can see very trend. We can see that their cell movement and muscle movement, many things we can detect it, then we could make the more specific type of a tissue. Okay, so this is the end of the slide, and that's all I've got today, and really appreciate it, and I just wanted to talk with you continuously. Thank you so much.
Thank you very much. And now we just finished all the talks from the international speakers, and we would like to get the time of the Q&A. So, do we have chairs in front and for the speakers? So we may have a 5 minutes break, the bio break. Yeah. So probably we can have a break until 5 o'clock. Yeah.
Okay. Can I ask the speakers to sit in front, please? All right. I know it's getting late, but we'll try to finish this by 5:30. Today. Today. All right. All right. Do we have everyone? Who are we missing? Dr. Rasko. Okay, great. Good afternoon. And first, I would like to thank the sponsor, Korean Federation of Science and Technology Societies, for inviting the world leaders in one place. And I would also like to thank them for personally bringing all my friends and colleagues in one place. So I'll be, we'll try to be punctual. And so I have two questions that I would like to ask each of the speakers. One is, given that your technology is, advanced and innovative, where do you think your technology will be in 5 years or 10 years? Okay. And then the second question to that first question is, what are the challenges that you envision? Okay, bringing that technology to where you want that to be. Third question of the first question is how can we overcome those challenges to get to where we need to be? Okay, that's first question, and I will give each speaker 3 minutes, no more. Now, it's like an open book test, right? Okay, now the second question that I would like you to think about between your responses is, nobody can work alone these days. You got to work together. So collaboration is, very important and very critical to the success of your, scientific career. All right. So what do you think the most effective collaboration method is? And if, let's say, Korean scientists or organizations or industry want to work with you and contribute to what you're doing, how could we make that happen? All right? I will give 2 minutes for that question for each of you. All right. Professor O'Connell, let's start with you. Only 2 questions.
Well, cell treatment, this field, we don't have an ideal system. Therefore, when I start to treat a patient using oral mucosal cells, make cell seed, and treat cornea for patient, start from 2000, almost 25 years ago. And 4 patients, we treated 4 patients, and we had a good results. And I got lots of information, telephone and letter from world, tell you have to treat a patient. And I go to government, and they gave me 80 difficult questions. And linear tumoralization, of course, important. Many, many important questions. But science, we cannot respond everything. But now, this society is moving, and we have many good bases, and therefore we can treat another patient in a different area. And already we treated many, many patients and autologous cells. But I think to expand this field, I think allogenic treatment, allogenic cell seed is important. I believe that's why I opened a new center in the University of Utah. And tomorrow I'll talk about the allogenic cell seed. And so future, we have to make a basic technology. And importantly, also system, we have to make system. To confirm the efficacy, we need a good clinicians, hospital. And to confirm the safety, we need basic science and government we need a good system, basic science and hospital, and government a kind of division. That's a future. Also, we need a new circumstance to educate new type of medical doctor and engineering. So we don't have an ideal position without like this fusion circumstance, new educational system, and the new type of medical doctor, new type of engineer is important. That I'm thinking.
Great, perfect. All right, Dr. Rasko, what about gene therapy?
It's a intriguing question to reflect on in terms of gene therapy. Some have similar experience to Professor Okano. I started the first clinical infusion of human gene therapy for hemophilia in 2001. We published our first reports in collaboration with the group in Children's Hospital of Philadelphia in 2006-2007. It was one of the first reports in the world. And since that time, we have seen approvals worldwide. So I think you asked a question, where will we be in 5 years? I think there will be an avalanche of new approvals for other gene therapies worldwide. Currently, we have 129 gene therapies approved in various jurisdictions worldwide. I think that number will probably double in the next 5 years. A lot of it will be CAR T cells, but a lot of it will be other orphan diseases and rare diseases. So I think that's one prediction for 5 years. You can invite me back and test me. The other one I will say, and it is in harmony also with Professor Okano in terms of the trajectory over the next 5 years about rationalising gene therapies. At the moment, it takes a minimum of 10 years to get from the first clinical trial to the point of approval. We cannot tolerate that any longer. It's just not fair. If you have 5,000 diseases and each one takes 10 years, we'll be doing this for hundreds of years. We haven't got the time, and nor have our patients got the time to be waiting for that. So something needs to be rationalised in terms of the current technology for example, ex vivo gene therapy, where we have to take the cells out of the body, genetically modify them, usually with a virus. That's typical now. You know, when I was a student, maybe electroporation or plasmids or other technologies that we use every day in the lab looked like they would work clinically, but I think that's really a long way off, if ever. But what I do think is that we will be moving to in vivo cellular reprogramming. So I think it's just crazy that a patient has to wait 6-8 weeks or longer for their CAR T cells. Soon we will have the ability to reprogram cells in the body, just inject the genetic payload, use very clever virus gene transfer technologies to target the specific cells that we want, whether antibody-mediated, small molecule-mediated, or simply using virus capsids to target particular tissues. That's a technology that should come sooner rather than later, because currently the cost of manufacturing cells like CAR T cells ex vivo or gene modification of hematopoietic stem progenitor cells ex vivo, it's a massive cost, a massive delay, and pretty much now we have the technology to be able to do it in vivo, just inject the virus-mediated gene transfer, or indeed, hopefully, the ultimate trajectory will be to replace all virus-mediated technology, because I would love to be out of a job. I've spent my entire career using viruses to facilitate the transfer. Please, everybody, switch off your mobile phones and I will be out of a job because I've spent my decades of research, my professional career, optimizing virus-mediated gene transfer. It's the safest, it's the most efficient. But I hope with the COVID revolution of companies like BioNTech and Nobel Prize-winning lipid nanoparticles, I hope that now we will be able to transfer mRNAs and even DNAs using lipid nanoparticles with a targeting capacity in vivo for in vivo gene modification. So finally, where do I hope there is opportunities for synergy? Well, I think it is also crazy that each country has its own systems of patents, its own systems of regulation. 80% or 90% of all regulation is harmonious in the world. We don't need different rules for different countries, and yet every company that wants to make a technology, sometimes for rare or even ultra-rare diseases, needs to go one by one, each country by country, to get regulatory approvals. So I think for many years, as president of the International Society for Cell and Gene Therapy and as an investigator, I've hoped that regulators would internationally come together and find a reason to harmonize so that we have a universality of regulation. So a company makes a curative therapy and can say, okay, now it should be available worldwide. There should not be these delays or these obstructions to such things. And finally, I think I will say that we need to find ways of getting lower costs, because we sit here in very economically advanced country like Korea, like Australia, like so many of the countries that we come from, sitting here, and we are very lucky. We are so lucky to be able to imagine a $1 million therapy. I just think that the vast majority of human beings on this planet deserve better, and currently they are denied even the most basic of therapies. So I think ultimately we need patient advocates, we need ethical people worldwide to rationalize regulation and also to rationalize the cost by making such things available. Thank
Wonderful. I feel and share your frustration. Okay, Pam?
Thank you. Both great ideas. I'm going to start with part C of question 1 first, along the lines of what was just said. I think I agree with you 300% that getting the world together on this would be very, very, very important, because what happens is that, more restrictive countries, the scientists in those restrictive countries go to areas where they don't have as much control. And that is good and not good, right? Because you can, maybe get your human clinical trials done or get some human data, but there might not be proper regulatory rules and regulations in place to make sure that it's safe for the humans, and also that we can get the key data that we need to come out of it at the other end. So I think that would be amazing. Lord knows what that would look like, though, trying to get all of the regulatory, in the States, we're just trying to think about how to get the FDA and the NIH together as far as like what animal models do we need to submit data to the FDA, and are those even valid animal models? Nobody wants to be working with beagle dogs. Everybody, or companion animals, terminal dog studies or terminal companion animal studies. So then what are the viable animal models that we can use? And that would be FDA, approvable, or do we have to move to ex vivo models? And we could do that if that's, if we worked on those and got those better. As far as my own technology in the next 5 years, so we started, here's even worse, we started on whole tooth tissue engineering. Our first publication was back in 2002. So that's 22 years ago now. And that was kind of using what Sang-Jin on the end, that Sang-Jin was talking about, although we were putting together postnatal dental epithelial and dental mesenchymal cells, not embryonic, and trying to, and seeded them onto scaffolds and trying to get
what, for you, the next 20 years.
Say it again? The next 20 years?
Instead of 5 years.
Yeah, so maybe...
No, I'm just kidding.
Maybe, no, it's true. So maybe, to be honest, we have a product called Regenagel. That is the first product in our company called Regenadent Incorporated, which I co-founded with Louise Bertassoni of Oregon Health Sciences University back in March of 2022. And we're submitting this for FDA approval in April 2025. And this is a cell-free dental pulp tissue regeneration product that forms a dentin bridge and pulp tissue, a dentin bridge in 5 days, and then dental pulp tissue and thicker dentin bridge that doesn't invade the whole cavity within 70 days. And so we have 2 large dog study models for that. We're hoping, and was suggested by the FDA to not do another large animal study because dog teeth are nothing like human teeth anyway, but to take those data and go to a small first-in-human, get permission, to go to a small first-in-human study. And so I believe in the next 5 years we're going to have a product. We're hoping to have an FDA-approved product in early 2026. And we have potential investors, industrial partners that are interested in exclusive manufacturing and distribution, et cetera, et cetera. So things are coming along. As far as whole tooth tissue engineering, my bet is on biodegradable microparticle cell mixtures, and maybe I can talk about this tomorrow a little bit, and Sang-Jin and I have been working on this for a number of years, for a number of years, where you can combine cells, or maybe some of these cell-free technologies that I was mentioned earlier today, like exosomal vesicles or metabolic signaling pathways that we can harness to try to get this to happen. So I think that there are a lot of new, really amazing technologies that continue to come along. Nothing has slowed down, right? Since this tissue engineering field began 40 years ago now, 35 something. It hasn't slowed down. It's only gotten faster and better. So I have a lot of hope and excitement about what can be happening.
Wonderful, thank you. Sang-Jin.
Okay, probably me.
Whoever.
So when 3D printing introduced in the field, we thought that it could be, it is basically automated process. So you can imagine that if you click that one button, then everything could be moving smoothly, and then you can finally have the construct that can be implanted in the body. But it is not really true because you need many components in order to achieve that goal. Not just only hardware, you need biomaterials and cell source, and you need to better understanding about biology and anatomy so that you can design more effective the construct that can eventually become a tissues. So next 5 or 10 years, I hope that there are many tissue construct developed from bioprinting technology, but probably it could be very simple tissue like bone and cartilage and skin. If there is not too much hurdle with that regulation. But if we talk about organ printing, it may be a little challenging to move forward because it requires a very fine resolution in order to creating a very small, tiny functional structures. And also, if you think about that, the kidney, so I'm not sure how many cell type in the kidney. So there are still many cell type is not fully identified. So there are another challenging to creating functional solid organ structures. And even though we printed very fine structure like microvasculature or nephron in the kidney, but we are not sure that when we implant in the body, how they integrate with host tissues, right? Even though you are creating very fine structure in vitro. So still there are many questions, but since technology is developed very fast than our expectation always, we may have some promising outcome within that period. That's I hope.
Great, thank you. Professor Igami, you're second generation. In 5 years, you're going to have third generation?
I think that, yes, there is like a 2.5, 3.5, 4, as I said, like a spiral generation transfer or exploration is essential, as long as it is a domain technology, yeah. And I think that, if we wait 100 years, innovation happen, but we want to accelerate it. So that accelerate means that a sort of like a biased. We cannot bring such an innovative environment for all of us. We have to select some and really support it to be diffused. But by the success of such a leading domain technology-oriented medicine, then that it gives it establishes a good base for other like related or technologies or in the people follow. So I was thinking about that like Amgen, when they really initiated a new concept enzyme treatment, U.S. government really supported them. And Amgen, though that they are now appealing their success story, but they had several times for like a bankruptcy, almost bankrupt. And actually, Kirin Brewery, the Japanese beer company, decided to work together with them. Yeah, and actually, Dr. Shimosaka, my friend, that he really discussed with Amgen and gave a lot of support, human resource to make them happen. And also that, actually we are like 3 physician scientists of Tokyo Women Medical University were sent to Amgen to help their product to come to market. So, so this means that, cross-border collaboration between such a strong science-based venture and big global company so big global company means that they have fund, global network, experience of business, and also like a power to negotiate government, not only one country, but other country. These, support is really essential to bring our technology from bench to bedside in the world, I think. So the key is that, how to incubate and select and incubate such a leader project, and let us work together to bring to the market. So actually on November 1st, I think I have to talk 30 minutes on your question. So I am preparing some slides, but I guess that not only academia, but we need to listen why the business company cannot step in this field with more significant size of fund or human resource than others. What is the hurdles? And let's take it out, and then at best, and find one like a leading project to bring to the world. I guess this may be the way.
Yeah, I think we will have, more time in our session 5. So let's see, Sangjin? Sangjin, okay.
Okay, so let me talk shortly. So in my case, I'm mostly nowadays working for the dental stem cells and therapeutic tool. As a therapeutic tool, I just wanted to try to inject in the body, not even oral area or the organ area. But biggest hurdle is the regulation actually, because this is a stem cell derived from the patient own. However, it just expand outside and then turn it back again. This is not same. We can't say, because already these cells got some the other nutrient from the FBS or some the other things. So actual technologies are a lot. We already developed a lot of technology and some resource materials, and also some scientists are very, very super highly outstanding. But one of the biggest hurdle is making the match with the government and clinical view and research scientists. Research scientists actually very superior, working very well, and many publication, many patent technology every day, every time is developed. However, in the clinical view, actual clinicians do not want to make a much risk, because if small things happen, patients gonna be died. This is one of the biggest hurdles. That's the reason why we actually were working with the clinician very well, like you. But this is a research and real clinic, this is a totally different story. But government says, I'm just saying about the regulation, which means if we develop some technology, such like stem cells, some kind of nutrients, some or another resource should be stocked in the outside, or some insurance issue. Also, there's a lot of things. So this is because I'm not senior, I don't know very well. However, I just wanted to know more and more. But I believe that 5 years or 10 years, this is, I think, the biggest problem and issue in my stem cell transplantation field. That's all I've got.
Okay, great, thank you. Yeah, I was hoping that we could come back this way, but we're running out of time. So, however, the purpose of this meeting is to address how to engage Korean scientists or organizations into, the advanced technologies, so that we can all work together. So let's see, Dr. Roscoe, I volunteer you. So what would be, how would you go about, and what would be a best way to do that? Let's say if there is a student or postdoc who wants to get engaged in your research activities, and get into your field. rather than tissue engineering. What would you do, and how would you advise that individual? Okay.
Usually it's a process. Obviously, we all get a lot of inquiries from optimistic young people, early career researchers and PhD students and postdocs. And mentoring is a fundamental part of all of our lives, seeing that the next generation is well trained and prepared for what the future brings. And what I love about it is that, we may spend years and years of our lives bringing something to the clinic, bringing something new, but for individuals who are just joining the clinic at the time or who've just joined, they take everything for granted when they see it. So it's just wonderful, you say, oh, well, can I explain to you gene therapy and how you can get a gene into a cell? And they go, oh, yeah, we know all of that. We got that at undergraduate. And so building the necessary skills and abilities for the next generation is crucial if we are to anticipate all of the problems that we are talking about here and now today, because those problems will be different. It's entirely possible, people are speculating that artificial intelligence will be smarter than human beings in 5 years' time, so that literally human species has never had another species on the planet that is smarter than us. But there is a real possibility that there will be singularity and that there will be intelligences, not just the difference between us and a chimpanzee, but if 5 times, 10 times up the scale of evolution, and it can happen so fast. We're not ready for that. So I think that in explaining how the next generation will cope with an ever-accelerating series of challenges, I suspect we're not, each of us, very well equipped, because we are ourselves dealing with these things on a day-to-day, week-to-week and month-to-month basis. It's awesome what even just natural language has changed our lives in summarizing papers. I don't know whether we'll be competing with each other, publishing papers even in a few years' time, because AI writes papers almost as good as human beings. So where will be the engine room? Where will be the motivation? I think these are real challenges and we're not ready for them yet. So I'm hopeful for the future. I'm, as you know, always enthusiastic and optimistic.
You are.
A glass half full guy rather than a glass half empty guy. But boy, the challenges seem to be enormous. And I think that maybe we haven't anticipated these surprises that are
I totally agree. And so getting back to my question, so if a student contacts you, you're open to, oops.
That's probably
Okay.
a student trying to contact you right now.
Oh, you're open to it, right?
Yeah, I think
Okay.
everybody's open to it. But the other thing is you just don't have a capacity in one lab to take on everybody. You want to help as many people as possible, but at the same time, the amount of effort in a 3-year, 4-year, 5-year PhD, it's extraordinary. You want those people each to succeed. You want to mentor them for the rest of their lives. What they sometimes don't appreciate is they come to you after a lecture and say, I'd like to do a master's or a PhD or a postdoc with you. And you know that it's almost like a marriage. It's longer than most marriages because they will be asking you for recommendations for their job in 10, 15 years' time.
Absolutely. I tell every candidate it's a 5-year marriage.
Yeah, minimum.
Yeah, so
But
we got to be
sometimes
careful.
they write back to you in 15 years' time. So, I know I haven't spoken to you for a long time, but can you write me a recommendation and remind me what I studied 15 years ago? It's happening to me.
How about a marriage without commitment? Okay. All right.
My wife would
Thank you so
approve.
much. I would like to thank all the speakers. Incredible talks. It was very enjoyable. And thank you all for staying till the end. But most importantly, I would like to thank the organizing team. I was watching it on the sideline, and there's so much work that went into this. So thank you for everything you did. Okay.
Thank you very much. But we still need to take a pictures all the speakers in front. It's just yeah. So you may stand up and in front, and then we took a picture together.